So first, we're going to go with the genetics fundamentals. So this is like the basic terms, so that hopefully it will lay the foundation for the rest of the sections that we go over today. So some main genetics terms that I think most should know is what the definition of a gene is, which is the basic unit of heredity, which is transferred from a parent to offspring. And a genotype is the genetic constitution of an individual organism. A phenotype is the observable characteristics of an individual. The phenotype is what you see. So like genotype is really what I guess distinguishes each organism from each other. But phenotype, sometimes the phenotype does not necessarily, I guess, the exact genotype is not exactly always expressed in a sense on the phenotype, just because, you know, there are some cases where there are dominant alleles, and in which case those dominant alleles will be seen when you are observing the traits of an individual, and they won't see some certain aspects like a recessive allele that's in it. And so speaking of dominant allele and recessive alleles, dominant alleles are an allele that masks the other allele in an organism. And a recessive allele requires 2 copies for the phenotype to be expressed. So as you can see in this chart to the right, usually for a gene, there are 2 alleles. Sometimes they may be 2 recessive alleles, so the 2 little g's here. Sometimes there may be 2 dominant alleles, which would be 2 of the big G's. And then sometimes they're heterozygous, which would be a mixture of that big G and little g. And this is kind of what I was mentioning before, but like when you have a phenotype, you're only seeing that trait that's expressed. So for this big G and little g, you would only see that it would end up becoming green because that dominant allele masks the other one, the small g. Okay, and then to go on to zygosity, I mentioned heterozygous. So homozygous is when an organism has the same 2 alleles of a gene, while heterozygous is when an organism has 2 different alleles of a gene. So homozygous would be either the 2 big G's from before or 2 little g's from before, and heterozygous again is the big G and little g combination. And hemizygous is when an organism has only 1 member of a chromosome pair or chromosome segment rather than the usual 2. So this is especially seen in gametes. So usually, you know, females have 2 X chromosomes, and then males have 1 X and 1 Y. So then in that case, males would be hemizygous for the X chromosome genes. Just a side note as well, all somatic genes are either going to be homozygous or heterozygous. Usually it's not the case that would be hemizygous. Those usually refer to the X chromosome. And then just a diagram again to the right. So these are like the alleles. So in this case, parent A has a dominant allele and a recessive allele. And then parent B also has a dominant and a recessive allele. And it just goes to show here that, you know, when one parent contributes a dominant one and another contributes a dominant one, you have different phenotypes based on the different combinations of the individual alleles. And now we're going to go into Gregory Mendel, who was the person who discovered, you know, these unique proportions of traits, and really was, I guess, one of the people who contributed a lot to genetics. So Gregory Mendel was an Austrian monk who had an interest in botany, chemistry, plant physiology, mathematics, and more. So he had a very large background in all these different studies. And his experimental organism was the garden pea, also known as Pisum sativum. And because of his background, he brought an interesting perspective on how to analyze what he was looking at through his garden peas. And so using the garden pea itself allowed him the ability to cross-fertilize the peas from brushing pollen from one plant onto a female organ of another plant, right? Because usually the peas and generally flowers have both male and female parts, so they are usually able to self-fertilize, but he decided to take parts or the pollen from one plant in order to do that crossing between each of the plants that he was studying. And also another really great aspect about him using the garden pea was that it had a really short generation time and could produce a large number of offspring, which really allowed him to do these experiments on the garden peas in such a short time. And also he was able to, observe a lot of change. Because if you're comparing that to another mammal of some sort, it would take quite a lot of time in gestation, and they probably wouldn't be producing as much offspring as a plant. And so that's what made it a really ideal experimental organism. And he also, in his experiments, tested clear-cut traits. So specifically, he did purple versus white flowers, yellow versus green peas. Later, when he continued doing more experiments with his peas, he learned a lot of other traits, which, it all contributed to his proportions and his discoveries. And so the other thing was that all of the peas that he used started with pure breeding lines, which means that they were constant from one generation to the next. And the way he did this was he really controlled his experiment, and he controlled the environment all of these peas were in to make sure that all of them started with, specific traits that they've always had. And that's how, when he was doing crosses later on, he could know that it was really because of, the mixing of traits or anything like that that caused the results that he was seeing from his flowers and peas. And so here on the right is a picture of him, and then also these are just some traits. So the flower color, white versus violet red, also referred to as purple usually in textbooks, and also pod. So this is just a extra trait, the full pod versus the constricted, and then as I mentioned with the color, the yellow and the green. And so another thing I wanted to mention is when he was observing, so there is a benefit of observing clear-cut traits because that's something you can easily see, as opposed to a continuous trait, which would be something like height, which takes a long time to also observe. So all of these factors played into how he made his next discoveries. And so the major laws that he determined was the law of segregation, which is when the two alleles from each trait separate or segregate from each parent during the formation of a gamete and then randomly unite at fertilization. And so I have this diagram to the right, which demonstrates this. So he determined the law of segregation based on his observations from his monohybrid crosses. And don't worry, this term I will go into in the next slide. And basically from meiosis, he determined and observed that, the row here, the chromosomes, each segregate from each of the parents. And so you see there's the R and Y and R and Y, the big one, the big R and Y on the right side. And then as they go here, they also go to the RY, and they just segregate so that there's one copy in each. And then eventually, once they form the zygote, they're uniting at fertilization. And then the second law was the law of independent assortment, which during gamete formation, the different pairs of alleles segregated independently from each other. And this he actually learned from his dihybrid crosses. It was because when he was doing the dihybrid crosses that has two different gene traits. I will also go over this term in the next few slides, but he noticed that the proportions were the same as when he did a monohybrid cross for like each of the traits themselves, in which case he knew that in that case, each of these traits were, segregating independently of each other and not depending on the other one. Okay, and then as time went on, although Mendel observed this specifically in his pea plants, there are many other exceptions and extensions to Mendel's laws, which have been determined over time. And so one of them is incomplete dominance. And so this is when the hybrid offspring of a hybrid cross does not have a phenotype matching either of the parents. And so one of the very popular examples that they do for, I guess, genetics classes or textbooks is that they have this red flower and this white flower that cross, and then they create this pink offspring. And so as you can see, the pink does not match either the red or the white completely, but instead it's like a mixture of both. And that's why, with Mendel's laws, he only determined like one or the other of the original parent generation. And so this is why this is one exception. And then the other one is codominance, which is when the hybrid offspring of a hybrid cross has both of the phenotypes from the parents. So we see here there's a two W's would lead to this white cow, and then this R and R would lead to this brownish colored cow. And then once you cross each of them, you see that both the white and the brown are displayed on the cow when they have both the R and the W alleles. So as you can see, both of these phenotypes are expressed, and that's why in this case it's codominance. I think another popular example used in textbooks is the spotted and dotted lentils. So spotted as one phenotype and dotted as another phenotype, and when you also cross those, the resulting offspring have both the spotted and dotted. And another exception that I wanted to mention was with blood types. And so actually so in addition to just like having two alleles that we've been kind of thinking about for each of the traits, blood type is really unique because it has three alleles. It has like the A allele, the B allele, and also the I, which if you have both of the I's, then it would lead to an O blood type. So that's another exception. And then more exceptions are seen in penetrance and expressivity. So penetrance is the proportion of individuals with a particular genotype who show that expected phenotype. So usually complete penetrance is considered when 100% of these individuals show that expected phenotype. It's incomplete when you're expecting the phenotype and less than 100% are showing that phenotype. And that example I was showing on the left bottom diagram here. So, the big C crossed with the little c and the little c. All of the offsprings here we know is the big C, little c. And we see that they're all supposed to express this gradient color. Now, we see that 4 of them do and 1 of them don't. And in this case, this shows incomplete penetrance because the phenotypic trait did not get expressed in all of the offspring. And then the second term is expressivity, which is the degree or intensity with which a particular genotype is expressed in a phenotype. So a popular example is with retinoblastoma. Some individuals have it affecting one eye, some have both, some don't have it affecting any eyes. So it's really like variable as to how the phenotype is expressed. And so this example I was showing in the bottom right-hand corner, where we see here the big C's and the little c's again, and all of the offspring have the same phenotype. Sorry, not phenotype, the same genotype. But as you can see, they have variable phenotypes. So they have this original darker gradient to lighter gradient. And here, and also like the darker gradient is more than halfway of the original oval. For here, you see it's pretty much similar to the parent, but as you keep going to each of these different offspring, it becomes less and less and less. So really, it's sometimes you cannot predict the expressivity of a certain phenotype. And so I just wanted to go into a question. So I'm going to give you guys 2 minutes. 20% saying A, 40% saying B, and 40% saying C. On the bright side, we have 0% saying I have no idea, so that's good. I am just going to reveal the answer. So the answer is B. And so this I'm going to explain a little more. So it says, the question says, a purple flower and a white flower are crossed. The offspring are a mixture of purple, white, and lavender. Each of these offspring have the same genotypes within their color groups, but different genotypes between color groups. Based on this information, which of the following terms best explains how the lavender flowers arose? And so, as we know, so we have 3 colors, purple, white, and lavender. And so the question says, each of the offspring have the same genotypes within their color groups. So all of the purple should have the same genotype, all of the white should have the same genotype, and all of the lavender should have the same genotype. But different genotypes between color groups. So that means purple, white, and lavender each have different genotypes across each other, right? And so we're trying to figure out how the lavender flowers arose. And as you can kind of see, purple and white, when those two mix, they kind of create a lighter purple. And so that's how we result in that lavender color. And so that most, corresponds, if you remember, to the red and white flower mixing to have that pink color. But also because incomplete dominance, it shows that, or the lavender genotype should have a mixture of like the purple and white alleles. And so having those two alleles mixed and not having a trait that's either one of them completely denotes here incomplete dominance. So a mixture of both. For codominance, it would require both the purple and the white to be shown. So it would be more like purple dots on top of that white flower background. And then for penetrance, they would all have to have the same genotype, but showing different not being expressed in all of them. So in penetrance, it would be more like if the purple and white flower, so the purple here and white flower are crossed, and then what would happen would be that, let's say the heterozygous, if we were using like a capital P and a lowercase p, those two should show like a purple offspring if they were both like completely dominant. And then what would happen would be then you would have like, let's say 4 expected offspring that have that big P, little p, and are supposed to show purple, and then only 3 of them show purple instead of all 4. So that would be if it was penetrance. And then thankfully no one chose Mendel's law of segregation, because lavender flowers and having this incomplete dominance is an exception to Mendel's original laws. And so that's why